afternoon, ladies and gentlemen, professors, associate professors, officials related to Pertanian, members of the public, fellow doctors, researchers, and scientists. I'm very privileged to have your audience today, and I'd like to talk to you about specifically on microbes for agriculture. And we have been trying this product in Kedong Jati and also in Klaten here in central Java. As an introduction, from an Islamic point of view, I have translated Surah Al-Arpudo into English, but I'm sure you understand what I'm trying to get at. The most important sentence here is, and we split the earth in fragments. Okay, so actually the microbes that I am dealing with are very many, up to 30 species. They come from nature, they are God-given. I did not invent them, I did not develop them. Certainly I did not genetically enhance them or genetically alter them. In America, where agricultural microbes are quite established, they have come up with a statement saying that we should be producing more food with fewer resources. Not many people believe that, but we have plenty of partners, trillions of partners. These are the microbes that help us achieve this goal. These are the uses of the microbes in a bigger, in an overview. And I must remind you that conventional agricultural practices have diminished the ability of microbes and diminished the ability of the soil to support plants to a satisfactory level. Again, plenty of benefits from microbes. These are broad category subheadings. I will also draw your attention to disease suppression and prophylaxis, which we have proven in Kedong Jati and we are now proving again in Klaten. You will have all this in your notes, but I will just emphasize on some of the points. Of course, everyone likes to have increased income. We like to have a reduced risk, and we also like to reduce our costs. This is me in Kedah, in northern Malaysia, on marginal land where they have paddy. And we have produced higher yields compared to weaker plants. This is marginal land. You call it S3, I call it C-grade land. Very sandy, very low colloid. Problems with drainage, or rather the other way around, problems with water, holding in the water. So we have produced significantly higher yields from that in paddy. The product has 30 different types of microbes. I'm going to segment them into these categories. Okay, and this is just a sample of the product seen in the bottle that we have been using in Kedong Jati and Klaten. For here, it is very important to recognize that we have signaling microbes. And why we are different from other products is because of these individuals here. I will remind you of the symbiosis, and this is a very important part of why our product should work in most soils. We are using symbiosis to our advantage. We are asking the plants will always give signals to the microbes in the soil. The microbes will respond to what the plants do by changing the soil here and pumping it back into the plants. So the microbes actually work very, very closely with the plants. And the autoregulation takes place between plants and microbes and the soil. Okay, stop there. The microbial action diagram. We have, like I say, 5 groups of microbes. Starting with A, the simplest ones, will help to fix oxygen, carbon dioxide, to some extent nitrogen in the soil. They will then help to sustain the next group of our microbes that have more work to do. They will be the ones that are breaking down your pupuk, your fertilizer. Okay? And they go on to the next ones, which are more complex. These are the ones that will break down your fertilizers, your manures, your organic compounds in the soil, crop residues, chemicals, poisons, a few other complex compounds in the soil. Next. Okay. Now, in combination of all this A, B, and C taking place, we have the disease-suppressing microbes that are over there in D that help to control a lot of the diseases that are found in the soil and on the plants. Apart from this, the greenhouse gas effect can be seen here. We are able to break down or use up some of the greenhouse gases to this effect. Coming back now to C. The group C composed of very complex microbes that do complex work in the soil and sometimes in the plant, and they will produce what the plant requires within the plant or within the soil. So your proteins, your growth promoters, your amino acids, your hormones, your complex carbohydrates are all the work, or mostly the work, of microbes that help the plant to build up its tissues, its biomass, its fruits, its flowers, its grain filling. Okay? Now coming back, the most important part is signaling microbes E. This group controls the activities of all of the other groups. So this is what we have also in our bottle. And this is the thing that controls and auto-regulates how much nutrition, macro or micronutrition, is going into the plant, depending on what the plant requires at what phase of its development. Okay? Now, I'm not going to go through point by point because this is very heavy. It is very heavy, but you have it in your notes. This represents function and effect of our microbes in our bottle. This was actually the target for year 2030 set by the American Microbiologists Association. They don't believe that anyone, even in their own country, can achieve this, but we have done that. We have done that in this bottle. So we are actually, technically, we are about 18 years or maybe 16 years ahead of our time. But it's good to see that the product is doing good work in Kedonggati and Klaten. Again, the same thing. Take the time to read it later on. Okay, one mechanism, very important mechanism, where our signaling microbes dictate what has to be done in the soil or for the plant. This is the structure of the mechanism. Auto-inducers and chemical signals are presented by the plant roots to the microbes. The signaling microbes will then govern the genetic expression and symbiotic competence. And that, in a way, it determines the motility, dormancy, sporulation, activation, multi-level functions of the plant, and resistance and anti-pathogenic functions. Okay. This is a typical population profile in the soil for one of our microbes, but certainly different microbes will present a different pattern. So you have a growth phase, which is a log signal induction phase, and then a plateau phase where the populations are quite high. And then they either run out of food or they run out of signals, so they tend to decline, but very slowly, in what we call a temporal shift phase. And then we have either dormancy or death. Okay. Next. Dormancy is very important because in our bottle, we will have microbes that are sleeping. In the soil, we also have the microbes that are sleeping, but they wake up depending on what they can do, what they cannot do, what are the stress levels, and even under starvation, they have a certain impact. Okay? The sustaining microbes do have a role, and it's quite a lot. They also work along with saprophytes in the soil. They work with desalinators in the soil. Now, how our microbes survive in the soil is up to the carbon-nitrogen levels, temperature, ferrite components, salinity. There's also competitive index where we compete against other soil pathogens. And there's a saprophytic activity. That means how much are we involved in saprophytic activity? How much does other saprophytic activity affects the microbes? Golden rule here. About 3.2 times 10 to the power 7 is 32 million microbes should be applied to 1 square meter of soil in 1 round of spraying. This is a rough rule of thumb. Disease control, very big topic. These are the various groups that various pathways of activity for disease control. It's highly scientific, but I'm sure you understand some parts of it. But what is most important here is environmental remediation, where we cure the soil of rotting components, okay? And we also reduce or selectively control the uptake of some of the poisons in the plants. We also have an anti-nematode capability, and these are some of the pathogens that are influenced by our microbes and certainly controlled by our microbes. Again, we have silicon soluble enzyme microbes. They have many functions. You will see this on your paper. Okay. Nitrogen managing microbes. In all the red dots, this is where we are involved in the nitrogen cycle. We are highly involved in the nitrogen cycle. Okay. And just for completeness sake, we also have the phosphorus soluble enzyme microbes. We select our microbes very carefully. We stick to the traditional microbes, and then we go on to the new generation microbes. Now, this is a photograph in Kedung Jati for corn. In this case, Pioneer 27, Jagong. Day 60 to 65. Showing first of all, this is a plant previously affected with bullae that has recovered. It was affected by bullae on day 15 to day 17, and it has recovered without any treatment for phosphoric acid or any antifungals. You will see one main corn, which is the apical corn, and then there's evidence of the subapical corn down here that's developing as well. So it has enough nutrition to produce 2 crops of corn. What is more important here is that the leaf that has been affected by bullae, the bullae has since been controlled. It does not spread. The leaf returns to health, but there is a remaining scar. And our microbes have penetrated the plant to provide this area, this zone of demarcation, which is very important. It means that the bullae is actively being controlled by our microbes also within the plant, and not just based on a contact capability. This is another plant also in the same area, showing that typically they do not get more than 1 tonggol, but now they have 2. So you have a subapical development. You have very nice fat stems, fat leaves, high photosynthetic capability, right? and overall general health on marginal lands in Kedong Jati. This is my version of the explanation for what happens in the soil. I won't go into it too much detail because this has been presented earlier on. Okay, next, go into this. If we have insufficient microbes from good soils, they do become poor soils. But there are other reasons for this. What we do have is in good soils, we have a balance of what is bioavailable and bio-semi-available, or what we call exchangeable cations and locked up cations. In poor soils, we have less of the bioavailable and exchangeable cations, and a very high amount of unavailable nutrients. If we use microbes, we tend to move towards that direction, which means that we have excellent soils, more is bioavailable to the plant, and there's a huge reservoir of exchangeable or semi-available nutrients. And we'd like to have an excellent soil category, okay? So the microbes tend to push things, they tend to push the balance towards that direction. Next. Environmental and soil remediation in broad categories, very important. This was covered earlier on as well, but we do all this, okay? Next. Vegetative effects on the plants, manifold. This is all within the physiological limits. Reproductive patterns as well as grain filling, these are all optimized. Okay, next. Okay, just to say that look, we have some results from rice studies. Again in Kedah, in Malaysia, the average yield on grade B land is about 8.4 metric tons per hectare. We have increased the yield to over 10 metric tons per hectare. But in actual fact, we could have done about 13. We could have hit 13 metric tons theoretically based on our scientific sample collections and data gathering on the field. It's just that we do have losses from the harvesting process, as well as from weather. So generally we are seeing about 20% increase in the yields of the paddy. Here, what is more interesting here is that this number for the variety of 220 paddy, we are getting almost 36 grams per 1,000 weight grains, 1,000 grain weights. Upper limit being 30, so we are actually about 20% higher for the 1,000 grain weight. This is all theoretical, all right? You can read through it again in your own time, okay? I'll show you this very simply. It's lima perut, and improvements have been seen within 4 days. New stem growth within 4 days. Extension of stem growth and leaf formation within 4 days. Okay, next. This is a kelapa sawit in a nursery. This has got microbes. This one is controlled without microbes after 8 months in the nursery. You will see here that no extension of feeder roots at the bottom, meaning that the plant has been able to get all its nutrition from just the soil that is inside the poly bag with the help of some fertilizers and microbes. Next. Okay. Okay, this is ladang durian tunggal, and we are seeing increases in yield. Very high increases. It's not usually we're hitting about 60 or 70% increases in the yield down here, all right, for year on year. Usually we hit about 30%, that's to be expected, but we're hitting about 69, 70%. This is very important because we do not change, again, I must remind you, we do not change the physiology of the plant. However, here shows a very high all to branch ratio percentage. The normal is about 24. or 22% to 24%. So we have optimized, what we have done is to optimize the physiology of the plant to perform at its best, given normal agricultural practices. Okay. Black bunch count, for those who know about kelapa sawit, okay, higher. Again, some numbers, very much along the same lines. Again, a high oil to bunch ratio count. Okay, this one, sorry, last one. This one is very important. For oil palm, there is an optimum level of NPK and a few of the cations in the oil palm. This is where evidence of signaling microbes, we are hitting the average, that means the middle part, what we call the bullseye effect, the middle part of each of the optimum range. We are hitting those numbers. And this is the work of signaling microbes, all this. To be able to manage the plant's physiology to its optimum in conjunction with average soil. Okay, next. Yeah, okay, just follow through. These are just more pictures, that's all. Yeah. Carry on. Some more. Yeah, some more. Some more. Okay, some more. Okay. This is analysis of the plant, of the soil, improvements in the soil, improvements in nitrogen and carbon, CN ratio is improved, cation exchange capacity has improved by 9%. Okay. Plant chemistry, variable, but again within some physiological limits. Next. Biomass. 52% increase, 24% increase on dry weights. Hydration, 9%. So we do produce plants that are a bit more hydrated. They are more drought resistant. Okay. These are just some front measurements, normal agricultural practice for oil palm. Okay, next. Spray application, very common. How we apply the microbes. EFB composting, sorry. Yeah. EFB composting may become the way forward, one of the ways forward for organic, natural ways of increasing soil sustainability and productivity from the soils. So this is what we do using our microbes, using very low-tech methods, okay, for treating shredded EFBs to produce good. These are early thermophilic formations. This is actually normal, not microscopy. It's actually normal photographs. Okay, close up. Early colonization for EFBs. This would be about day 15. The broad range of microbes that are doing their work at the thermophilic phase. You see about 8 species are involved in this. Temperature is about 65 to 70 degrees core. This is mesophilic, which is the midline, middle range curing of the compost. And you can still see some colonies of microbes. Silica crystallization also takes place at this time and breakdown of microtubules. Okay, final product is this on day 65. Okay, just some scientific numbers for the quality of our compost. The NPKs, the CECs, the CN ratios, etc. Okay, Kada, Padi trials here. Okay. Microbes. Okay, microbes have been applied here. Less trip infestation, mouthpart insects will decrease. Okay, 63% increase in yield. We use Rilong. This is on marginal land, and they're getting about 7 metric tons per hectare on average. Compared to 4 metric tons on average. Right. This is one of our records. We have got about over 15 metric tons per hectare in Sekinchan in West Malaysia. This is quite interesting because we have a lot of stiffness in the plants, we have healthy plants and very good grain yields. These are what we could expect from increments from 20% to about 30 up to 52% increments, but the average increments in yield is about 30%. This is more rice data. Okay, keep going. Okay, so I just leave you with this thought that we can use marginal land to improve your yields. It's not just keeping the yields the same, it's to improve them. Thank you very much.